for the conferences. She successfully completed the government research projects funded by the Madhya Pradesh Council of Science and Technology. She extended her contribution to deposit new culture in the various culture collection centers. She is awarded and felicitated for a number of best presentations, poster presentation, and so many awards during her path along the research as a researcher, as a scientist. She delivered the invited talks in the various national and international conferences. Under her research guidance, 2 research scholars completed their PhD degree and 2 are working to pursue the PhD degree. She also works as a PhD thesis evaluator. She is working as a subject expert for the biotechnology and microbiology for the Swayam Mass open online courses by the Ministry of Human Resource Development and develop the e-contents in respect subjects. She was as a member of a board of studies microbiology Devi Ahilya Vishwavidyalaya Indore. She also worked as a member of an examination committee and a member of a board of studies from the Devi Ahilya Vishwavidyalaya Indore. She developed the industrial consultancy with a number of industrial sectors like Pharma Limited, Indore, and the Natural Care Private Limited, and Research Center, Indore. She extended the research consultancy in also with various research centers in the MP. She is a reviewer of a research journals, the Journal of Advanced Research and the South African Journal of Botany. She extended her contribution to organize a number of conferences, webinars, training programs, and so many academic events. Such a versatile, dynamic, knowledgeable personality is with us today. I request Professor Sheetal Bhashin to guide us on antibacteria, nature's maintenance machinery for revival of environment health. Now, I will to this environment doctor to guide us. Welcome, ma'am.
Thank you so much, ma'am. Am I audible to you all?
Yeah, ma'am, you're audible.
Okay. Thank you so much for the introduction, ma'am. First of all, I thank all the organizers, principal ma'am, the entire management committee, and all the team members of the national conference for inviting me for a talk here and helping me to present my views in this conference. Thanks a million to all of you, ma'am. Can I share my slides, please?
Yes, sir. Yes, ma'am.
Okay. Are the slides visible, ma'am?
Yes, it's visible.
Okay, thank you so much. So let us start with the nature's maintenance machinery for revival of environmental health. Basically, I'm talking about Actinobacteria. Actinobacteria, they are a special class of bacteria which resemble fungus in their growth pattern, but they are basically the prokaryotes, exactly the bacteria which grow in diverse environmental niches. So moving ahead, the thrust of my talk will be on the solutions rather than the problems. All of us have gathered here to talk about the environmental issues, the upcoming burning problems which the environment is facing, and definitely it is because of human beings. All of us know about all the different types of problems. My concern will be talking about the solutions, how we can solve these problems. So my first slide is on the basic tools and techniques which we will follow to solve these problems. So the actinobacteria or we can call these organisms as actinomycetes also. They were earlier known as actinomycetes because their growth pattern resembles fungus. But basically they are bacteria, so now they've been termed as actinobacteria. So what kind of maintenance machinery these actinobacteria have to solve the problems related to the environmental issues? First of all, the recycling of organic matter, basically by composting all the organic material which is present in the form of animals and plant remains. Then mineralization of the compounds. Ma'am, can you see my slides? Is my second slide visible or still the first one is visible? Hello, ma'am.
Hello, ma'am. No, only your first slide is visible.
Okay, I'll just change it. Just a minute.
And please make a slideshow.
That is what I was asking. Just a minute. Can you see the slides now?
Yeah, the same slides will now slideshow.
Is it still not in slideshow?
No.
Okay, just a minute. Just a minute.
No. You are sharing is you stop sharing.
Yeah, I'm just doing it. Yes, now can you see my second slide?
Yes, ma'am. Okay. Yes, good.
And
Yeah, we
now
can view the show.
are the slides moving?
Yeah.
Okay, okay. So I was talking about the maintenance machinery. First, we'll talk about the recycling of the organic matter, then the mineralization of the compounds, revitalization of soil fertility, degradation of dyes, degradation of pesticides, and bioremediation of heavy metals. As I told you, that we will be concentrating on the solutions. First, we'll understand what exactly my organism is going to do. Yeah, what exactly my organism is doing for solving this problem. Then we'll come to what exactly the problems are, right? So before we understand how this organism solves the problems, we must know what these organisms are and how are they into solving the problems. Because nature is a beautiful creation of God, and God has set each and everything in its place. It is only for us to understand what exactly is to be used where. There is no need of human intervention to solve problems. God has created diverse creatures which can maintain the entire universe on its own. We are still in the process of understanding what exactly nature is doing to balance the complete ecosystem. And whatever I'm trying to present here is only a very, very, very small part of the automatic machinery which nature has already created. So we will be doing this work with the help of actinobacteria, and these are a few representatives of actinobacteria, the microorganisms which we are growing in our laboratory. All these pictures are basically from the cultures, from the organisms we have isolated in our own laboratory. And first of all, I thank all my students who have helped me doing this work, my lots of MSc students, BSc students, and my PhD students who are working under my guidance on this field on actinobacteria. Some of the cultures I've isolated during my research work, and some of the organisms my students have isolated. This is the way we grow our cultures on petri dishes. These are different types of actinobacterial isolates which are growing on plates. This is a special kind of a technique where we isolate a single bacterium from a mixed culture and study on the basic characteristics of those organisms. So now coming to little bit of those problems which we must discuss. The basic cause of pollution, there are two major causes, the natural causes and the anthropogenic causes. You must be surprised that I'm saying there are natural causes also for pollution. There are lots of natural causes for pollution. Like, for example, if you talk about the heavy metal pollution, the weathering of rocks, the volcanic eruptions, and the climatic changes which are happening on the entire globe, they contribute to some of the natural pollutions. And the anthropogenic pollutions, which we talk about majorly, they are the industry-based pollutions, the agricultural reasons, and the urbanization. And the major thrust today we'll be having on the pollutions caused by industries. They are by tanneries, textile mills, metallurgy, galvanizing factories, distilleries, pesticide industries, fertilizers, paints, varnishes, and pharmaceutical companies, to name a few. But there are some more industries which release some harmful chemicals in the environment, which we have to deal and which we have to find solutions for. So, a brief introduction to the organisms. This picture is just a relative picture to understand the size. The uppermost picture, you can see the hair, the human hair, and look at the size of the organism as compared to the human hair. This green colored ball which you can see is the ragweed pollen, and you can see the red blood cell, and you can see a very tiny small bacterial cells in the bottom of the picture. So we'll be talking about such kind of organisms in detail now. Coming back to the Actinobacteria, they are also known as ray fungus. Why are they known as ray fungus? Because they resemble fungus. Their growth pattern resembles fungus. Initially, they were described by Kohn in 1875, long back. But their major, they came into light majorly in 1943 because of Selman Waksman. Waksman is the person who really made these organisms important. He discovered streptomycin, the antibiotic streptomycin, from Streptomyces griseus. After that, many scientists were, after finding different strains of actinomycetes, working on different strains of actinomycetes, isolating these organisms from soil. Now they are gram-positive bacteria and they have high GC content. The most important thing is they are very slow growers. E. coli grows on petri dishes within 24 hours, but this organism takes 3 to 4 days to grow on petri dishes. But if they are in pure culture, they might grow in 48 hours, and then you can you can get the peak growth of the organism in 5 days. But when they are in mixed culture, when you want to isolate it from the nature, from soil or water, anywhere, it will take minimum 4 to 5 days to appear as colonies on the plates. The pictures which you can see on these slides, they are of the organisms which we have isolated in our laboratory. They are also considered as the highly evolved, most evolved bacteria, because they have a very long gene pool. They have a very high genome size. They have great genetic and functional diversity. Why am I saying that they have great genetic and functional diversity? Because they produce millions of different types of bioactive compounds. We'll talk about them later. They have great functional diversity because they produce millions of types of enzymes and surfactants and many things which we can use for solving the problems of our environment. The diversity in the production of secondary metabolites is also very large. You all understand what are secondary metabolites, and these metabolites are produced in the stationary phase of the growth of the organism, and they have the major contribution, their genome size, the highest contribution of their genome, their gene pool on the secondary metabolome. By remediation of pesticides, toxic metals, and radioactive waste, these organisms are very, very much available for degradation of your pesticides, the toxic metals, the heavy metals which are polluting our water bodies and soil around us, and the radioactive wastes. There is a strong correlation between the multiple antibiotic resistance and heavy metal resistance. Now, what exactly is this? When basically actinobacteria are famous for the production of antibiotics. Around 70 to 75% of the antibiotics which are available in the market, they are produced by actinobacteria. And among this category, streptomycetes are the ones which produce major part of the antibiotics. Now, when the organism is producing an antibiotic, it will definitely itself be resistant towards that antibiotic, because when it is producing that antibiotic in nature or in a monoculture as we do it in the laboratory, it has to be strong enough to survive while producing such an antibiotic. So many of these organisms are resistant to lots of antibiotics which they are producing on their own. And all these organisms who are resistant to the antibiotics which they are producing, they are also resistant to the heavy metals in the environment. And we will be encashing this property of the microorganisms to bioremediate the soil and water samples which are contaminated with heavy metals. Then they are also good at removing the xenobiotic compounds. What are xenobiotic compounds? The recalcitrant compounds, the compounds or the materials which are very difficult to degrade and remove from nature. Now, if you talk about the diversity of these organisms, you'll find these organisms in nature in unicellular form also and in filamentous form also. This slide which you can see here on the right hand side is the microscopic picture of actinobacteria, especially the streptomycetes. The streptomycetes are the organisms which are majorly present in the environment. They are present in the largest number in the soil. When you try and isolate these organisms from nature, you'll first of all pick up streptomycetes only. They appear as white colonies on the petroplate in the beginning, and later on they appear as gray colonies. And if you look at them in the microscope, you'll see Maggie noodle type of an appearance. Can you see this picture? The filaments are the mycelium, the substrate and the aerial mycelium, and these spring-like structures are the spore patterns. They are the beads, the small spores arranged one after the other in a spiral pattern. If you see a spiral pattern of an actinobacteria under the microscope, you can be sure that these are streptomycetes. They are, again, talking about the diversity, they are one of the highly useful ones because they're producing lots of antibiotics. Then the bifidobacterium, this is also in the category of actinobacteria, which is used as a probiotic. Then on one side, they are highly useful, and on the other side, they are highly pathogenic also, like mycobacterium, which causes the tuberculosis. They are aerobic also, they are anaerobic also, they are microaerophilic also, depending on what kind of nature, what kind of habitat they are growing in. You can find these organisms in the extreme environments also, high temperatures, very low temperatures, alkalophilic nature, acidic nature, all the natures, all the environments, you'll find these organisms. Then talking about the classification, this is an ever-growing group. The group is growing slowly and steadily. Earlier, they were confined to only a few orders and a few families. Now, they have around 300 species, and the number is still growing. Again, the pictures which you can see on the right-hand side, they are actinobacteria, which we have grown in our laboratory, and every organism is producing some or the other important bioactive compound. Then They are universally distributed, and the distribution definitely depends on the season also. If you talk about spring seasons, 20% of the microbial flora is Actinobacteria, and in autumn it is around 30%, and in winter it reduces to 13% because of the frost in the environment. They are the organisms behind the earthy odor of soil. You must have enjoyed the beautiful earthy odor, the fragrance of soil after the first shower. That is because of geosmins, and that is a volatile compound which is produced by Actinobacteria. They are present in soil, fresh water, hot springs, marine environment, salt lakes, invertebrates, plants, everywhere these organisms are present. Now, what is special about these organisms, and why am I using these organisms as solutions for the environmental issues? So we'll start that. The first thing is that they have the largest genome size among bacteria. I'm talking about bacteria. So among bacteria, they have a very large genome that is around 8 megabases, which is around double than a typical free living bacteria. For example, E. coli's genome size is around 4 megabases, somewhere around 4 to 4.5, and some strains have around 5 megabases. So because they have a large genome size, they have a very large genome size concentrating only for the secondary metabolome, the secondary metabolites. And these secondary metabolites, they are contributing to the, I mean, this portion of the gene is contributing to the production of antibiotics or pigments. They are the extraordinary source of lead structures. What do you mean by the lead structures? All of us are finding out new microorganisms which can produce antibiotics against the multi-drug resistant bacteria. Lots of pathogenic organisms have become resistant to the uh, antibiotics which are already available in the market. What will happen after a few years? We'll not have any antibiotic to treat infections, to treat the pathogens, to treat the patients. So people are looking for new antibiotics, new organisms which can produce these antibiotics. This is the largest reserve which can give you more new types of antibiotics, and that is the lead structure. So this is an extraordinary source for the natural products. And as I've told you, that streptomycetes share 80% of it. Then they have now it is observed that since last few years, earlier there were only classical methods to find out that which organism is producing which kind of an antibiotic. There were submerged fermentation technologies, there were solid state fermentation technologies, but now with the help of bioinformatics, we are able to find out that how many genes these organisms have for the production of new chemical entity. Now, what is this new chemical entity? It is basically the new compound which we do not know of, but it is there, it is present in the organism. The organism can produce this new compound, and we are not able to synthesize it. So we are finding ways and means to make the organism produce this new compound. This is the actinobacterial life cycle. We'll just move through it. They are the filamentous organisms, and they produce spores. These spores falls on the substratum, and then they start reproducing just like any other filamentous organism. These are few cultures of the laboratory. Then again, coming back to the basic point from where we understand why these organisms are so important. They have a large genome size of around 7.7 to 9.7 megabase pairs, and the streptomycetes genome is most worked upon. they have lots of genes which can produce antibiotics or which can produce lots of enzymes which are present on the genome or on the plasmids, and they can be horizontally transmitted from one organism to another very easily. And one important point is the synteny. There exhibits synteny among lots of actinobacterial strains. Now, what exactly this means? You can see same compound being produced by lots of different types of organisms in this category. So synteny is conserved more in metabolic genes as compared to other genes. That means the metabolism, the genes which are responsible for producing the enzymes or the genes which are responsible for producing the antibiotics, they are flowing in the organisms generations after generations. But while these genes are flowing in generations, there is some bit of changes appearing at every stage. And this some bit of difference which is appearing at every stage is helping us getting new kind of bioactive molecules, new kind of bioactive substances which are helping in solving the problems. For example, the first actinobacterium, which was sequenced fully, like all of us are scared of microorganisms, that microorganisms are very harmful. But let me tell you that the organisms which we all know, the entire number of organisms which we know, only 1% of the microorganisms are harmful to us. Only 1%. Mind it, only 1% of the organisms are harmful to us, and rest of them, they are either useful or harmless. So as the human nature goes, when it was on sequencing the organisms, they sequenced the most dreadful organism, the Mycobacterium tuberculosis. Then they shifted on to the organisms which were useful, like Streptomyces silicola. This is an organism which produces lots of different types of antibiotics. Then the Streptomyces, Evermellitis, and then you have a long list which have long genome size ranging from 7 to 9 megabases. Now we come to the biosynthetic gene cluster. What I was telling you about the gene synteny, the organisms carry specific set of genes which are basically responsible for the production of antibiotics. These portions are known as the biosynthetic gene clusters. When we had earlier methods, the traditional methods of only doing maybe submerged cultures, fermentative production by microorganisms and producing antibiotics by submerged fermentation technology or by solid state fermentation technology, we used to know about only 4 or 5 different types of compounds which a microorganism used to make. I mean to say 4 or 5 different types of antibiotics which a microorganism used to make. But after the advent of bioinformatics tools, we've come to know that each Actinobacteria, each streptomycete is capable of producing more than one antibiotic, and the limit goes beyond 20. There are some Actinobacteria which can produce more than 20 different type of antibiotics. Now, these portions, these set of genes which are present in the organisms, they are known as biosynthetic gene cluster, which can biosynthesize different types of antibiotics. And the antibiotics which we are not able to produce in the submerged fermentation technology, and we are not able to make the organisms produce it, they are known as the cryptic biosynthetic gene clusters. That means the organism can produce these antibiotics, but we are not being able to make it produce in the culture. Now, there are different numbers like Streptomyces silicola has 23 such set of genes. That means you can understand 23 different types of antibiotics this organism can produce. Ivermectin can produce more than 29 different types of secondary metabolites, and you have a list of Salinispora can have more than 17. Rhodococcus have 31 potential secondary metabolic genes, the biosynthetic gene clusters, and Mycolotopsis has 29 cryptic gene clusters, means there are 29 more antibiotics which this organism can produce, and we are not able to do it. Scientists are finding methods, ways, and means to make the organism express these genes and to make the organism produce these antibiotics. Maybe these antibiotics will add to wonders, and one day we'll be able to solve all the problems and all the, cure all the patients who are suffering from the infections of multi-drug resistant organisms. Then talking about the secondary metabolism gene share. Why are we talking about these things for Actinobacteria? Only to understand how these organisms will be solving your problems. Which problems? The environmental issues. So the secondary metabolism gene share, if the organism has a very small genome size, maybe of 3 or 4 megabases, it cannot afford secondary metabolism gene share. There will be very, very less genes for secondary metabolism if the genome size is small. But the Actinobacteria have a large genome size, therefore, the secondary metabolism genes are also high in number in them. Around 5 to 10% share, which is the highest among the bacteria that is present in these organisms. As you can see, Salinospora has 10.9% of the genes. Then Salinispora tropica has 8.8% of the genes devoted for secondary metabolism. Streptomyces evermellitus has 6.43% of genes devoted for secondary metabolism. But the organisms, if they have the gene size less than 3 megabases, they will not have the secondary metabolism gene cluster. Now we come upon the problems. Till now we were discussing about the organisms which we will be using for solving these problems. Now we've understood the solutions, the organisms which will be helping these problems. The first one I'll be discussing will be the heavy metal pollution. We have lots of origins of natural and anthropogenic ways by which the water, soil, the sediments, the air, and the living organisms are polluted with heavy metals. The heavy metals have their own different type of negative effect on human beings, the animals, the vegetation. The excess accumulation of these metals in the entire globe is harming the health indirectly for humans. The basic ones are the chromium, arsenic, lead, cadmium, mercury, zinc, tungsten. All these heavy metals are accumulating. They are increasing in number in the environment, and they have lots of negative effects. For example, the lead poisoning leads to loss of appetite, hypertension, renal dysfunction, birth defects, fatigue, arthritis, lots of problems related to all the organs of the body. If mercury concentration increases, it can permeate across the biological membranes, and due to this, it can damage the nervous system, brain functions. It can lead to shyness or tremors or memory problems. Now, one important thing is that, okay, these heavy metals are present in the environment, and we are talking about remediation of these heavy metals using microorganisms. There are lots of microorganisms which can solve these problems. Now, how are they solving the problem? If we talk about the organic matter, if we talk about the animal remains or the plant remains, We understand that the microorganisms produce lots of enzymes and degrade those complex organic matter into simpler ones. All the complex organic matter is converted into carbon dioxide and water, and the complex matter is, it is all finished from the environment. It reduces the BOD and COD. But if you talk about heavy metals, where will it grow? Metals are the elements, the last stage. Where will it break down? Where will it lead to? Because if the organism is bioremediating it, what is it doing? Is it eating up? Now, we have to understand one logic, one mechanism, and that is these metals, all these metals are required by, they are present in the nature. If they are present in the nature, that means nature needs them. That means plants need them, humans need them, all the biological systems need them. So if you talk about chromium, chromium with oxidation state of 6 is highly toxic, but chromium with the oxidation state of 3, that is essential nutrient supplement. So you are using these organisms to convert these highly toxic state of metals to the useful states. You have to convert these heavy metals from a toxic state to a state where they are not harming the environment. So this process is a bit different from degrading the organic matter. So you have lots of microorganisms and also the actinobacteria which convert chromium from its toxic state, the chromium 6 state, to chromium 3 state. We will be converting the oxidation state, or we will be reducing the solubility. If the heavy metals are present in nature, if they are highly soluble, they will move into water, they will solubilize in water, move to different parts. They also move through air. They are reaching to different parts of the globe. From where they are originated, they reach to distant parts and contaminate the environment of distant parts. So if we make the heavy metals bio-unavailable, you must have studied bioavailability. If the substance is bioavailable, then only it will be able to act on any animal or plant cell. But if it is made bio-unavailable, or if it is converted into an insoluble form, its toxicity will be highly decreased. And that is what we are doing in bioremediation. So we'll be using Actinobacteria for this work. Heavy metals such as copper, lead, mercury, and some other metals also, they lead to schizophrenia and atherosclerosis, which are highly dangerous state of affairs for human beings. So conversion to less toxic form or immobilization in order to reduce their bioavailability, this is the aim. And the streptomycetes, we worked upon these metals, like we worked on lead, we worked on chromium, and we have found that streptomycetes and arthrobacter, they can bear very high concentrations of chromium and lead in the liquid state. The organisms which you can see on the right hand side, you can see the pictures of streptomycetes. These, as I told you, that they appear as filaments in the microscope, and the spore pattern appears as the spirals. These organisms were able to withstand high concentrations of chromium, high concentrations of lead. And first it has to withstand that much high concentration, then only it will be able to bioremediate. To remind you again, this picture downwards is the streptomycete colonies growing on a petri dish. And if you see these organisms under the microscope, you'll be able to see in this form. These are the scanning electron microscopic pictures of the cultures which we've isolated in the lab. So chromium 6 resistance has been observed in several Arthrobacter strains, and Frankia. Frankia is also one of the strains among Actinobacteria, one of the groups in Actinobacteria, which can bioremediate the heavy metals. They are able to survive in the soil and in water, which are contaminated with the heavy metals. Propionibacterium freudenrichi, Bifidobacterium, they are able to remove cadmium and lead toxicity. Acidimicrobium ferroxidins, this is an extremophile and this can survive at very low pH of 1.8 and can tolerate zinc up to very, very high levels. Then next issue, we move on towards the textile dye pollution. All the beautiful colors of our clothes, of our utensils, of plastic bottles, or whatever daily items which you use in house, the beautiful colors, they are polluting ultimately the environment also, because whatever amount of dyes are used for manufacturing in the industries, 10 to 15% is being wasted. Whatever amount of dye is being used for dyeing your clothes, around 12% of every company is throwing away the dyes in the effluents, the industrial effluents. The synthetic dyes are used in paper industry, textile industry, pharmaceuticals, and food industries everywhere. And significant amount is being discharged in the environment. And while going through the fields, while going from one city to another, if you go by road, you can just see lots of ponds, lots of pipelines draining sewage here and there. They are highly toxic, they are highly mutagenic and carcinogenic. All of us know about that. And there are lots of chemical methods for, degradation of these dyes. But those methods are again leading to some residues which are harming the environment. But if you talk about microorganisms, they will completely degrade those dyes to the components or to the state where the BOD and COD of the sewage is reduced to a very good extent. These dyes are usually degraded by a variety of enzymes produced by organisms like licases, the manganese peroxidase and lignin peroxidase. To name a few, there are some other enzymes also produced by microorganisms which can degrade these dyes. Like, for example, we've been working on Actinobacteria, which can degrade the dyes. You can see the pictures here. These pictures, these are the azo dyes which have been incorporated in the medium. The petri dish contains Evans blue and this petri dish contains Congo red. These are the azo dyes, and most of the dyes which are used at industrial level are azo dyes and they are highly contaminating. So the pictures which you can see here, the Actinobacteria is growing in the center and you can see a clear zone around this actinobacterial growth, which is showing that these organisms have degraded these dyes. Further, moving on to the secondary screening, where we have shown that this is again the Evans blue, which is present in a test tube, and these are the treated samples. We have treated these samples with actinobacterial cultures, and you can see the reduction in the color of dyes. So this is the way you can degrade the dyes. Okay, Actinobacteria is helping you degrade the dyes, solve the environmental issues, and then you want colors. Like you cannot survive without colors. So what to do? Have the natural colors. We can go in for natural colors. These organisms will provide you natural colors in the form of melanins. They are the brown colors. They can produce lots of other colors like blue colors, red colors. I'll show you. We have a long list of biocolors. We have a long list of pigments which are being produced by actinobacteria, the carotenoids, the uh, uh, protogenesins, the anthracyclines, the phenoxazoins. Yes, all these colors are producing All these organisms are producing different colors which you can use to dye your cloth, which you can use to incorporate your different articles, and you can use it in pharmaceutical preparations also. The most important part is these pigments have antibacterial activity also. There is a connection between the antibacterial activity by microorganisms and the pigment production by microorganisms. All the actinobacteria which are producing antibiotics, most of the actinobacteria which are producing antibiotics, they are also producing pigments. And these pigments can be used as food colors. If you want to have some green color in salads, you can use green pigments. I have that also. I'll give you that. See, here it is. On the right hand side, this is producing olive green pigment. You can use this. If you want chocolates, if you want to color your chocolates, you can have the brown pigment melanins. If you want to have if you want to prepare some pharmaceutical preparations like syrups for children, the syrups are colored, they are pink colored, they are green colored. You can have red or orange or pink pigments from these actinobacteria here. You can see. So there is no need. You want to have blue color, you can have blue color from this organism. So you name the color and you have them in the natural process by the organisms. So aren't we solving the issues? Aren't we solving the environmental issues? You can also contribute in solving the issues. This is a picture which is showing different types of pigments which are produced by actinobacteria. When the organism grows in liquid, it grows in this form, the beaded form. And you can see the dark color here, the melanin pigment dark color. Now, in the morning, Madam explained so beautifully about the pesticides. So my next point is working upon the pesticides. India is an agriculture dominated country and 60% of our land is used in agriculture. And as ma'am said, 50% of the Indian population is involved in agriculture. But we must know that there is a silent storm coming up in the form of these poisonous chemicals, the pesticides. They are definitely improving the product yield, but they are also causing huge problems to us. To jot down a few problems, you can have a look at this. These are disastrous problems which chemical pesticides are giving us. It damages the immune system, the neurological system, the reproductive systems, liver, kidney, abortions, infertility. They are having lots of problems in all the organs of our body. They are present in all the food which we are eating. They are present in milk, milk products, vegetables, fruits, even in breast milk also. So how to solve these problems? Again, we can have the organisms to solve these problems. There are lots of pesticide degrading actinobacteria, the Arthrobacter, Rhodococcus, Streptomyces, Frankia, Mycobacterium, Nocardia, Pseudonocardia, which are able to degrade these pesticides, and scientists have worked upon. They've been able to remove 73 to 75 and in some cases around 90% of the pesticide content in the samples. And as the organisms grow in groups in nature, so scientists are also using consortium. Consortium means you mix 2 to 3 different types of organisms together, and one organism is degrading one pesticide, leaving it to a certain level. The other organism is using that end part as a substrate and then degrading it to another level. That way, you can have a group of organisms which can completely degrade the mixture of pesticides which are present in the soil samples. Then, okay, The problem created by chemical compounds that can be cleared with the help of actinobacteria. And we are telling our farmers not to use chemicals as pesticides. So the solution, another solution is you can use these organisms as biocontrol agents. The best part is if you use these microorganisms as biocontrol agents, matlab, if you want to degrade, if you want to destroy the pathogen which is destroying the crop plants, the insects which is harming the crop plants, you can use actinobacteria which are pathogenic for these insects. That means the entomopathogens. They will be specific only to the insects which are causing harm to your crops. They will not kill the other insects which are present in the field, which are useful for the crops, which are useful for pollination, and we will not lead to the extinction of the other living systems, which will not lead to the extinction of other insects. The beautiful butterflies which madam showed in the morning, we have lost such beautiful butterflies because of use of excessive chemicals in the field. If you use these biocontrol agents, means if you use these actinobacteria to control the diseases in plants, they will only kill the pathogens. They are not going to harm the other insects or other plants. Similarly, one more thing, they are not going to get bioaccumulated if we eat these plants which are treated by actinobacteria. The remnants of the organisms, they are not going to cause harm to human beings because these actinobacteria are pathogenic for those specific pathogens of plants, for the phytopathogens, and not for humans. They are not causing disease to humans. Then this is another set of pictures which you can see. All these are fungus grown on petri dishes. The black color is the fungus, the aspergillus, which is growing on the petri dish. And here you can see that we've added the actinobacterial supernatant, the antifungal produced by the actinobacteria, and it is inhibiting the fungus on the plate. So these are the different phytopathogens on all the plates, different types of fungal pathogens which cause diseases in plants, and we've grown different types of actinobacteria which are inhibiting the fungal pathogens, the pathogens which are causing diseases in plants. These organisms, the production technology can be scaled up and can be applied in the field so that the chemicals are used less, and we can use these natural antibiotics produced by the organisms to solve the problem in the field. Then not only inhibiting the pathogens, not only inhibiting the phytopathogens, but if you apply these actinobacterial cultures in soil, it will be working as plant growth promoters also. They produce lots of plant growth hormones, because of which the productivity of plants increases. They help in fixing the nitrogen. They release indolacetic acid. They release gibberellins, cytokinins, which are important for plants to grow. Then one more important point, that is the soil washing technologies. You must have heard that there are oil spills, there are like places near the petrol pumps or places near the refineries or somewhere, sometimes accidents happen and the ships explode. The ships which were carrying crude oil, that explodes and that sinks, and the oil, the crude oil, that is overlaid on seas, which results in the death of lots of aquatic ecosystem animals. So how to get rid of that? There are biosurfactants being produced by actinobacteria, not only actinobacteria, other organisms also. And these biosurfactants help in removing that oil, that material from the surface of the ocean layers. The surface active molecules which are being produced by actinobacteria, they help in soil washing. What do you mean by that? there are certain pollutants which are present in soil and which can be treated, which can be removed if you apply microorganisms there. But these substances are not bioavailable for the microorganisms. So what to do now? If we add biosurfactants, they are the amphiphilic molecules, they will take away those substances which are attached to the soil particles. They will take away these things away from the soil particles and make it bioavailable for the microorganisms which you have added there for bioremediation. So some organisms like Nocardiopsis, Streptomyces, Amycolatopsis, these organisms have been worked upon to remove the heavy metal pollutants or other hydrocarbons from the soil particles and make it available, make it bioavailable for the bioremediating organisms. Then one more big problem, that is the production of paper. When we produce paper from paper pulp, the bleaching has to be done, and the amount of water which is used and the amount of chemicals which are used for bleaching of paper, they are highly carcinogenic and they are released in the environmental water. Without, if you leave it without treatment, it is going to cause excessive environmental hazards. But if you apply organisms which are producing lacases or which are producing xylanases, you can do the bio bleaching process. That means the color of the pulp, you want to brighten the paper pulp so that you get a bright colored paper. You can brighten the paper pulp with the help of these enzymes which are produced by organisms rather than the hazardous chemicals. So these are a few solutions which I proposed. This is my laboratory where we work with our students. This is my college, Maharaja Ranjit Singh College. And with this solution, with this bundle of solutions, the Actinobacteria, I thank you, and I'll try to solve the queries if you all have any. Over to you, ma'am. Can you hear me, ma'am?
Yes, madam.
Yes.
If anybody have any queries, then ask. The session is open for the discussion.
I acknowledge all the researchers, I acknowledge all the work of the scientists and industrialists from where I have taken all the data and all the pictures. All the pictures of actinomycetes are completely ours. Rest of the pictures from the industries and the effluents, they are from different researchers. So thank you, ma'am. If anybody has any question, I'm here to answer.
No question. There is no question, I think. So, because madam has explained the topic very nicely and clearly, every point we are able to understand about the Actinobacteria, measures maintenance machinery for the removal of environmental health. Madam started with the basics that if anyone don't know about the Actinobacteria, then he will also have to understand it, diversity, distribution, features, life cycle, biosynthetic G++, secondary metabolism, heavy metal pollution, bioremediation of heavy metals, textile dye pollution, biocolors, pesticides degraded by the Actinobacteria, means every role of the Actinobacteria which is performed to to the betterment of the environmental health is explained by the madam, and it shows that the madam deep knowledge and does own work with the confidence madam can explain it. So thank you very much, madam.
My pleasure, ma'am. My pleasure.
Validatory function. So please remain. Okay.
Yes, ma'am.
Immediately we are starting the validatory function.
Yes, ma'am.
Just feedback will be provided in the chat box during the validatory function. Now I call the Bhutada madam, Madhuri Bhutada madam, Mathematics department, our college, for the next validatory function. Sir, madam, bolo.
Namaskar and good afternoon to all. I am Madhuri Bhutada. It is a matter of immense pride to stand here before all of you this afternoon. It gives me a great pleasure to extend a very warm welcome on behalf of myself and from our institution. We are delighted to offer a gracious welcome to all of you. I welcome Honorable Dias, President of today's function, Dr. J.V. Dhanvit sir, and Convener Dr. J.S. Dhande, and Organizing Secretary Dr. M.S. Chaudhary madam. Since morning, we are here in one-day online national conference on recent trends in environmental and biological sciences, sponsored by Kaviyatri Bahinabai Chaudhary, North Maharashtra University, Jalgaon, and organized by Botany and Zoology Department of our college. Now, in the afternoon, we are here in the closing ceremony of this national conference. I request all the dignitaries and participants to accept my oral welcome. I welcome once again everyone present here offline and online. We have been listening to eminent speakers since morning. All these valuable insights give us a new perspective. Inviting assembly is the toughest job. Positive approach impress the mob. Coming together is success. Walking together is a great progress. Dr. J.S. Dhande is our convener. So to take review of today's national conference, I request Dr. J.S. Dhande, sir, for convener's note. Sir.
Good afternoon, friends. Now, we are coming toward the conclusion of this conference, and very happy as well as thankful to all the speakers, resource persons, as well as the participants who give a good response for this conference. We are in short period, but in short period, all of them give the good response. So, dear friends, Kavitri Bahinabai Chaudhary, North Maharashtra University, Jalgaon, sponsor one-day national conference has been organized by Department of Botany and Zoology of Srimati C.K. Koteja Mahila Mahadal Bhusawal, District Jalgaon, on topic recent trends in environment and biological sciences. Now today, in this conference, about 129 participants registered. Out of themselves, 77 participants will be present for this conference, including most of the part of these our country, that is like the state like Rajasthan, Bihar, Jammu, Kashmir, Uttar Pradesh, etc. During inaugural function, in an introductory speech, Dr. Meena Chaudhary, organized sector conference, explain the themes and objectives of the conference as well as represent the whole scenario of the college. Chief guest talk today's function as well as inaugurated of this conference. Dr. Kishore Pawar, Registrar Kavitri Bainabai Chaudhary, North Martin Jalgaon, state that today all of us facing huge problem regarding the biological resources which now existing. We must aware it and we must fight against the environmental conflict facing today. He gave best wishes for the conference. President of this function, our principal, Dr. Kala Sahab said, the college has sustainable resources up to a certain extent. We must create natural resources for the better future. This conference will be surely create awareness about the environment and the biosphere among the society. The opening ceremony was concluded with the vote of thanks by myself. In a keynote address, Dr. Meera Srivastava, Principal, Government College, Lunkansar, Bikaner, Rajasthan, delivered her presentation on topic pesticides, impact on environment, and botanicals as a safe alternative. In spite of using chemical pesticides, we must or should turn toward the botanicals, integrated pesticide management, and natural enemies, natural predators, pathogens, etc., so as it can be helped to minimize the chemical pollution. In plants, nearly such of these, the near about 350,000 compounds are present, which are isolated as a botanicals from the plants, and these have the great potential. Lastly, she told, we need good present and future generation, so always use green and eat green. In the morning session, second invited talk delivered by Professor Gita Padate Madam, Department of Zoology, Maharaja Sayajara University of Vadodara. She delivered speech. She explained a success of dominance of species. She focused on different era in geological scale. Science of technology developed first. She pay attention on ozone depletion and its danger, smoke, which is unhealthy to the human being. Also pay attention on climatic change. She gave the example of this house sparrow. We all know that house sparrow is a neighborhood, present in neighborhood, but now this sparrow is the species of sparrow decline not only by the environmental pollution, but due to the human civilization, because we not maintain the space for this sparrow. This sparrow built up their nest in our houses, but we do not have the such of these spaces for that for their building of nest. And so we must try to build such of these. We must try to give the such of this opportunity to the sparrow so that it can be increased in a future. During afternoon session, second invited talk by Professor Sitar Basin Madam, Head Department of Biosciences, Maharaja Ranjit Singh College of Professional Sciences, Indore, MP. She focused on the role of the actinomycete and Actinobacteria, which plays a key role in a biosphere. Nature is a beautiful creation of God. Antibacteria create genetic and functional diversity as a production of secondary metabolites. Bioremediation correlate with the multiple remove xenobiotic compound, etc. This It is a heterogeneous group. So she explained features of this bacteria as well as the bioremediation bacteria. By bacteria, bioremediation can be done of the heavy metals by bacteria like Frankia. In a textile, we can use the natural colors which are maybe manufactured in future by means of these different bacteria. We can use the Biocon by these actinomycetes. So this is of the today's. I think this is the conference may give the fruitful knowledge, and we may hope that this knowledge may be helpful in the future for the researchers as well as the botanists as well as zoologists. Thank you for your presence. Okay, thanks again.
President of today's validity function is Dr. J.V. Dhanvij, Vice Principal of our college. He is Head of Department of Computer Science and Physics, currently working as IQAC Coordinator and has impressive experience of teaching and research. In order to felicitate Honorable President of the conference, I would like to request convener Dr. J.S. Dhande sir to felicitate him. Thank you, sir. At its best, feedback is a powerful tool, stimulating insights and improvement. And so we take the feedback of delegates. So first of all, I would like to request Dr. S.B. Patil, sir, Sardar Vallabhbhai Patel College, Anpur, Maharashtra, to share his feedback. Sir, please. Dr. S.B. Patil, sir.
Okay. I really congratulate the organizing team of the college for organizing such a knowledgeful national level conference on the topic recent trends in environment and biological sciences. The resource person in two sessions in their keynote address enriched the knowledge about the role of various microorganisms, plants in the cleaning of the environmental health or maintaining the environment health. Dr. Meera Srivastava focus on the
To request you to please on your video. We are not visible, sir.
Dr. Meera Srivastava focus on the role of environment-friendly biopesticides as an alternative over the synthetic pesticides and their impact on the environmental health. Totally, the biopesticides are the eco-friendly pesticides which maintain the health of the environment. Later on, Professor Grita Patate enlightened on environment past, future, present, future, and focus on the various environmental hazards. Then afternoon session, Professor Sheetal Basin focus on the various genetic resources on the antibacteria and their role in the production of the secondary metabolites, bioremediation, bioleaching process of the heavy metals. So overall impact of this conference is very good and enrich our knowledge. So thank you giving to me you opportunity for express my views about the conference. Thank you.
Thank you, sir. I really appreciate the details you shared with us. Thank you once again. Now, I would like to call upon Dr. Sagar Dhangar, Faizpur College, Maharashtra, to share his feedback. Dr. Sagar Dhangar, sir. I think Dr. Sagar Dhankar sir is not here. So I would like to request if any of the participant wants to share his or her feedback, I would like to invite him or her. If you want to share your feedback, please unmute yourself. Uh, Now, Mr. Sanjay Verma wants to share his feedback with us. So I request him to share his feedback. Sir, please unmute yourself, sir. Sanjay Verma, sir.
Sorry, ma'am. Sorry, ma'am.
I think there is connectivity problem. Sir was interested, but because of the net problem, he may not be able to do. Now, we come to the next step. The sales of this particular notion were adjusted by a person who is not only a great motivation to all of us, but a visionary and a person who gave us the belief to bring together this event. He is none other than our beloved Vice Principal, Dr. J.V. Dhanvit sir. I cordially welcome our today's President, Dr. J.V. Dhanvit sir, for his presidential speech. Sir, please.
Good afternoon to everybody. First of all, I am thankful to Research and Development Section, Kavitri Bainabai Chaudhary, North Maharashtra University, Jalgaon, who sponsored this conference. And I am also thankful to botany and departments heads and their colleagues who choose such kind of interesting topics over here. And all the participants who are present for this conference, some participants from our state and some other participants from the other states. So here in my speech, I am focusing on the new trends in environment science. So what are the new trends? So there are 8 new trends here. I am focusing that. So the first trend is here, increase in renewable energy sources. So this is quite important nowadays. We know that the conventional sources of energy exhausted soon. And so we need to turn over to renewable energy sources. So we know that scientists trying to search the new devices and means here increasing the efficiency of that devices. Okay, so I think that if we motivate the peoples to use such kind of renewable sources, then I think we can overcome the problem faces nowadays. So in our college, I'll tell you the example. In our college, we installed 20 kilo kV solar power plant and street light lamps, solar water heaters everywhere. So our audit report comes, green audit report, environment audit report, and energy audit report. So our building declared as a zero energy building. So, and since last 3 years, from 1819 to 2021, our college building is a zero energy building. So through this conference, if we motivate the participants, so I think we can use the renewable energy sources. Okay, and we overcome the problems faced by the conventional energy sources. So other thing is reduction in energy consumption. So other trend is reduction in energy consumption. So actually what is happen? So we will find every home. So every person have a vehicle, every person have a mobile phone. So energy consumption increases day by day. Okay, so we have to overcome this problem. So if we turn over to the use of conventional, non-conventional energy sources, that is renewable sources of energy. So I think we overcome this problem. And suppose we motivate, we start to motivate the students from our college to the society, then I think we can reduce the energy consumption. I mean to say that the conventional energy sources consumption get reduced. Okay, so this is one of the problem. We have to use alternative renewable energy sources. Then this problem will be overcome. So third trend is now regenerative agriculture. So regenerative agriculture is farming strategy that priorities soil health, land management, and biodiversity during the cultivation and harvesting of food product. With regenerative agriculture, deliberate practices are put into practice that minimizes the loss of necessary soil nutrients and minerals, thereby preserving the life of the soil. Foods grown in these environments are termed regenerative organic products. This trend is expected to grow in more farmers become vested in maintaining soil health and vitality. So this is quite important to regenerative agriculture. Then fourth is the relationship to plastic. Most of the peoples are already
familiar with the dangers of plastic, such as the effect of pollution. So, sea pollution, so that is very dominant pollution nowadays, and difficulties in dangers over the past several years. There has been an increase in plastic recycling as well as the use of more biodegradable products. This trend towards greater plastic recycling and the use of biodegradable products is slated to further increase. This is especially the case as companies are making increasing efforts to recycle their plastic for reuse. So recycling and reuse are very important because plastic is very dangerous. It's produce the pollutions. Means water pollution, air pollution, environmental pollution. So we have to turn to recycle the plastic and then reuse. In this way, we can means overcome this problem also. Then fifth trends in environment science, human waste management. This is very important thing because over 2.4 billion people worldwide engage in poor hygiene and sanitary practices as a result of poor or lack of sanitation facilities, and mostly in means progressing countries or developing countries. So we will find this such type of problem mostly in India. So as a result, these individuals are prone to a whole host of waterborne illness, especially diarrhea-related deaths in children. However, due to greater awareness of these health hazards, measures have been implemented to provide adequate sanitation facilities for those at risk individuals. These measures are expected to increase and innovative solutions are developed and implemented. So if we means develop and implemented such practices, so I think this will be managed, human waste management we will do. Then next thing is the accelerated climate action. So this is also important as the effects of climate change become more apparent and devastating. Individuals and corporations are increasingly stepping up to take action to further halt or even reverse the damages that have already taken place. Previously established global partnership and alliances are expected to work together nowadays to develop and implement strategies to protect ecosystem and decarbonize the economy. So for doing this, we can accelerate the climate action. Then impact of cement. We know that India is a developing countries and nowadays road also made with the help of cement, and we know that cement emit the carbon dioxide, okay, carbon's product. So cement is a significant source of world carbon emission. It emits the carbons accounting for about 8% of the world total. As such, efforts are being made to curb carbon emissions from cement. Research is currently underway regarding new techniques and raw materials that can be used in making cement more eco-friendly by reducing the levels of carbon emitted. If we use the garbage or eco-friendly materials for making the cement, so then we can overcome these problems, cement problems. Then last thing is I'm focusing air travel and climate change. So air travel is a significant source of greenhouse gas emission that are harmful to the environment. Okay, it contributes about 2.5% of the global carbon emission due to air travels. So air travel is increases. Now means ordinary people or medium people also travel through the air, and due to this reason, what will happen? So carbon greenhouse gas emissions that is harmful, that is given out by the air. So given this grim statistic, research is underway to make aviations more energy efficient as well as eco-friendly. So though far from complete, strides have been made to using biofuels as alternative fuel source. If we use the fuel in air plane instead of that is conventional fuel, so we can use the biofuels. Okay. So additionally, research is ongoing in the development of electrical and solar power plants. Okay. Now, just like satellite, now satellite launching satellite, working of satellite on solar power plant basis. So similarly, such technology we can use in plane also. If this technology adopted, I think air travel and climate change, we can overcome these problems. So these are the issues nowadays we are facing and developing simultaneously here. So through this conference, we spread the message from student to stakeholders. I think this is the great success of this conference. Thank you very much.
Thank you, sir. Sir has concluded in a very simple and effective way. Sir, you advocated for green energy, zero energy, preservation of soil, regenerative agriculture, use of biodegradable products. You advised to focus on reuse and recycle of products and use of eco-friendly products, biofuels and solar energy for the welfare of whole mankind and world. For your motivational words and valuable guidance, once again, thank you very much, sir. Now, I would like to call upon Organizing Secretary of this conference, Dr. M.S. Chaudhary, Madam, to extend vote of thanks.
Thank you, madam. Good afternoon to everyone. We hope that you found the conference informative and worthwhile. I am here to provide vote of thanks. First of all, we are very much thankful to Kavitri Bahinabai Chaudhary, North Maharashtra University, Jalgaon, for giving sponsorship for this conference. Then, the chairman of this valedictory function, Dr. J.V. Dhanvi sir, Vice Principal of our college, we are thankful to them for guidance in this valedictory function. A special mention to our principal, Major Dr. Mangla Sabadra Madam, being the catalyst that inspired us to do out our best and stand as a pillar of power. We are also thankful to the resource persons of today's conference, Dr. Meera Srivastava Madam, Principal Government College, Lunkas Nasar, Bikaner, Rajasthan, Professor Geeta Pathe Madam, Department of Zoology, Maharaja Sayajirao University, Vadodara, Professor Sheetal Bhashin Madam, Head of Department of Biosciences, Maharaja Ranjit Singh College of Professional Sciences, Indore, MP. All the resource persons spent their busiest time gracing the occasion. Today, we had the opportunity to hear their thoughts, and this is definitely encouraging us in our future events. Your thoughts have enlightened our minds and shown us a new path. Thanks to all the speakers for ignoring this occasion and sharing their opinions today. Our sincere gratitude goes out to all the participants and well-wishers for your rock-solid support system and encouragement. Without your support, the conference not able to excel. Thank you very much for joining this event. And lastly, I am also thankful to all the teaching and non-teaching staff of our college for successful organization of this conference. Thank you very much.
Thank you, Madam. I declare the closing of this conference with the permission of organizing committee and honorable president of the valedictory function. The valedictory function is over. I am sure this program is knowledgeable feast to everyone who has participated. I extend my best wishes to everyone and once again, thanks to all. Dear participants, here important news for you. Everyone has to fill their feedback forms. It's compulsory to all, without which certificates not be issued. So please kindly fill the feedback form. Thank you. Thank you very much. The link provided in that box. The link is provided on a WhatsApp group. So kindly fill the feedback form without missing. Thank you.